Welcome to the end of our first week of this intersession and the last lecture that we're going to do on the action and mechanisms of proteins, specifically enzymes. After this, we'll probably look at some DNA replication. We'll look at protein synthesis, but we won't be talking about proteins as active molecules. We'll just be talking about how they end up being made, and then we'll switch gears and talk about energy production and photosynthesis towards the end. So this is kind of the end of a little section here. Okay, so before we really get into enzymes, there's a couple of things that I forgot to talk about last time. So one of those are going to be types of enzymes, and the other are little molecules called ribozymes. So actually proteins don't have a monopoly on acting as biological catalysts. There are RNA molecules that are capable of speeding up reactions. The most famous of these were discovered in the early 80s. They were studying excision of introns or something like that. In this, I don't remember where this was happening, what school or what college. But they were having a hard time finding which proteins were actually catalyzing the process of cutting out introns. If you don't know what introns are, we'll talk about them when we get to our DNA. But they're basically non-coding portions of a gene that are just kind of intermixed with our regular coding parts of our gene. And in our bodies, we have those little non-coding bits cut out before our proteins get made. So they were having a hard time figuring out what protein was doing this job, and as it turns out, it wasn't a protein at all. It was self-splicing RNA. And since then, there's been other examples of catalytic RNAs that have been found. Now, catalytic RNAs are known as ribozymes. And ribozymes are not rarities of nature. They are actually pretty prevalent and essential. Of the protein-making ribosomes that we have are basically giant ribozymes. In fact, the 23S ribosomal subunit of the prokaryote and the 28S ribosomal subunit in our bodies catalyze the formation of peptide bonds. So ribosomes are important in our understanding of the evolution of life. If you remember, the first chapter in our book talks about how nucleic acids were not only storing the genetic information, but capable of catalyzing their own replication. And so that actually answers one of those chicken and egg questions. Which came first, enzymes that do the work of a cell, or nucleic acids that carry the information to make those enzymes? And as both the carriers of the genetic information and as the catalysts, the ribozymes are likely both the chicken and the egg in the origin of life. Okay, so some of the things that I wanted to talk about last time also were the 6 major groups or classifications of enzymes. So I've got a few different examples here for us to look at. So the first of these 6 is the oxidoreductases. These are obviously oxidation or reduction reactions, and in our situations in biochemistry, this usually means the addition or the removal of some hydrogens and electrons. And so this pyruvate into lactate is an oxidation of pyruvate, and I'm sorry, the wrong way, is a reduction of pyruvate and an oxidation of NADH. So this is the one being reduced, and this guy oxidized. Again, in any enzyme that catalyzes this, and these usually can go both ways as well, would be an oxidoreductase. Now there are transferases. These just transfer groups, so they form new covalent bonds. They don't use energy to do this though. You can see here aspartate and alpha ketoglutarate. This aspartate aminotransferase will take an amino group from the aspartate, put it onto the alpha ketoglutarate, making glutamate, which is a precursor to many of our amino acids, and then oxaloacetate. So that's transferase, and there are all kinds of transferases. We've got a hydrolase. Hydrolases basically cut with water, so they hydrolyze reactions. Here we have acetylcholine reacting with water. Acetylcholine esterase here is the enzyme that's the hydrolase, and then it cuts the molecule into 2 bits. We've got lyases. Now, lyases add groups to double bonds or remove groups to create double bonds. So again, adding or removing groups. And here you can see an example of a lyase down here. This one is adding a water molecule to this double bond here, and you can see it gets added on, hydrated. Okay, isomerases. Isomerases, like they sound, isomerize reactions. So the molecular formula remains the same, it just moves some things around. In this case, alpha D-glucose 6-phosphate gets converted into alpha D-fructose 6-phosphate. We go from a 6-membered ring into a 5-membered ring. And lastly, there are ligases. Ligases are the ones that require ATP energy, and they connect molecules together or form new covalent bonds. You can see here L-tyrosine and the tRNA that would carry L-tyrosine get added together or connected via tyrosine tRNA synthetase. And this requires ATP. But again, these are the 6 major classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Okay, I should have left myself a little bit of room to draw. That's okay. Let me see. Let me jump back here. All right, so let's talk about allosteric enzymes. So we need to remember from the previous lecture, what was it, yesterday, that there were 2 types of enzymes. We saw when we did the curve of the chymotrypsin, and it was at ATCase or something like that. That chymotrypsin actually behaved like this, and ATCase was something a little more sigmoidal. Sorry about the lack of being able to draw some sigmoidals there. But if you can kind of remember that, and it indicated 2 things. One was that the sigmoidal shape was an allosteric enzyme, and the other one was not. So allosteric enzymes, allosteric deriving from the Greek, which means other shape. Allosteric enzymes have biological activity that's affected by other substances binding to it. This alters the conformation of the quaternary structure. Now there are allosteric effectors, and we'll talk about a few different kinds, that are substances basically that modify the behavior of an allosteric enzyme by binding to it. And we said that ATCase, or to ATCase, the aspartate transcarbamylase, the one that we looked at last time, was an allosteric enzyme, and it actually has it's an allosteric enzyme that's involved in feedback inhibition. So we're going to talk about what that is in a second. We saw that ATCase and hemoglobin exhibited cooperative effects, and so we're going to explore a little bit more about that when we look at how some of those subunits can change and then influence the increased binding or the decreased binding. We'll see how that sigmoidal curve sort of happens. And then we'll look at how allosteric enzymes have different responses to the presence of inhibitors that are different from non-allosteric enzymes. So the first thing is the feedback inhibition. And so actually I probably could have just stayed on this slide here. So you can see here that feedback inhibition basically means that some process in which the final product of a series of reactions inhibits one of the first reactions in the series. And so aspartate and carbamoyl phosphate, you see here, ATCase is involved in creating carbamoyl aspartate. Now then carbamoyl aspartate goes through a series of reactions where it ultimately generates CTP, and CTP is able to actually inhibit this enzyme. So high levels of CTP indicate that we don't need to keep doing this reaction, and so that's going to go and actually put an inhibiting effect on ATCase. And again, you can see in this second sort of image here on the right that there could be many different enzyme steps, many different parts of this reaction, but the whole point of feedback inhibition is that one of the final products, if not the final product, goes and stops an early enzyme in the series. It doesn't necessarily have to be the very first reaction, but it could be one of the early reactions. Okay, we saw that ATC ACE catalyzes the condensation of aspartate and carbamoyl phosphate to form carbamoyl aspartate, and we saw that there was a sigmoidal curve that describes the allosteric behavior. And so this graph that we're looking at here is showing the velocity versus the substrate. Of course, here the substrate is aspartate. There were two substrates, but so this is just for aspartate as the substrate. And they kept the carbamoyl phosphate levels constant for this. So what we can see here is a comparison between the effects of the inhibitors and the activators on the regular sort of motion, or the regular activity, I should say, of this enzyme. It's easy to see that there is differences. So activators like ATP will actually cause this to be less sigmoidal and actually more hyperbolic, like we saw with non-allosteric enzymes. Sort of just kind of turns off that non-allosteric part. Turns off that allosteric regulation and kind of just says, do your job, go. Whereas the inhibitor we saw goes in there and actually stops this from being as active, and we'll look at how the inhibitor actually does that task in a few slides. Let's see. And again, CTP, remember, is that final product in the line. So this is the feedback inhibition. The inhibitor actually goes and shuts that down pretty fast. Now, down here I've got the ATC ACE that we can look at. It's actually made up of a few different subunits. There's the catalytic subunit, shown in green. 6 proteins organized into 2 trimers. And then we've got the regulatory subunit, and this is 6 proteins organized into 3 dimers. They can actually separate these two from each other with treatment of, p-hydroxy mercury benzoate, I think it is. And this reacts with the cysteines in the protein and actually breaks it down. When we break it down and we just isolate the catalytic part of it, we see sigmoidal curve. And when we add ATP or CTP, we see no change. It doesn't get changed at all. So just removing these regulatory parts tells us a lot about where, we know that they're regulatory because when we remove them, we can't regulate it anymore. But what it means is that's where the CTP and the ATP are effectively binding. There's a couple of different systems we're going to talk about. Sorry, I jumped ahead in my slides too fast. There's 2 types of systems. There's K systems and there's V systems. K systems are enzymes for which the substrate concentration that yields the 1/2 of Vmax is actually altered by the presence of inhibitors or activators. And the V systems are the enzymes in which the presence of an inhibitor / activator changes the max velocity, but not the substrate level that yields 1/2 of Vmax. So they get designated by the 2 different types, whether Vmax changes or whether Km is affected. And they don't actually call it Km, though, for these systems because the Michaelis-Menten equations don't apply to the allosteric enzymes. They're just for the non-allosteric from the last chapter. I think they call it the K.5 or the KD or something like that. So it slightly means something different. The K.5 is the substrate level at 1/2 of Vmax in a K system. There's different types of allosteric effectors. So there's substrates that can be the effectors. There's inhibitors. There's the activators. All of these can bind and affect activity. There are homotropic effects and heterotropic effects. We touched on this briefly in an earlier lecture. But basically a homotropic effect is when several identical molecules can bind to a protein. This is like oxygen and hemoglobin. Or heterotropic effects. This is where different effects occur when different substances are bound. So the activation by ATP and inhibition by CTP are examples of heterotropic effects as far as ATC case goes. Now, let's talk about the concerted model. That's what these pictures are showing. So this concerted model is different than the sequential model that we talked about in a different lecture, where different subunits will change by being next to a subunit that gets bound to a molecule, right? When we were talking about hemoglobin, we saw that one subunit binds oxygen. We get a subtle change in that that tertiary structure that affects the neighboring subunits, and now they're more receptive to binding an oxygen, and then in turn they change and it binds the next one. That's sequential. This is concerted. This says that all proteins, whether or not they have multiple subunits, have two conformations. They're either in an active state, or designated R for relaxed. This binds a substrate tightly, or they are in an inactive state, tight or taut, however you want to think of the T, and this binds substrate less tightly. And so in the absence of substrate, most enzymes are going to be in the T state. And they actually came up with a ratio of the two states, the T state over the R state, and designated this L. And when L is really large, and L is usually really large, this just means that the T state is in the favored or unbound state, or has less affinity state. Of course, both of these are unbound, but T is less likely to bind. Now, the presence of substrate will shift the equilibrium from the T state into the R state. In changing from the T to the R state and vice versa, all the subunits change simultaneously. So that's what we're seeing here. If the substrate, even when the substrate binds, of course, let's talk about what that means first, though. So I said two things. Let's talk about the first thing. Presence of substrate shifts the equilibrium. So here substrate is being added. Think of this as three different things. This is R state, this is T state, and this is RS state. Okay, they're all different, and they all have their own equilibria. RS is not in equilibria with T. R is. So when R starts to be converted into RS, now there's less R, right? There's some amount of R that was present. Now it's being turned into RS, and so there's less R. That means the equilibrium between R and T shifts, and T starts to convert into more R to reestablish equilibrium. This is how binding of the substrate to the R, to the active form, actually shifts the equilibrium to there be more active form. This is kind of how cooperativity works. Now, in that T form changing to the R form, this T form has two subunits. They both turn into the relaxed at the same time. So here you see the different sites. They're changing shape at the same time, both subunits, this subunit and the second subunit. And again, this is just the concerted model of this. Now, we can expand this a little more and talk about what happens when an activator or inhibitor binds. So let's look at that. So the binding of the substrate induces a conformational change in the T form to the R form, and we can see that here. So this part of it's still the same. This whole little branch of it is still the same as what we looked at previously. Now, the R form is actually favored by the allosteric activator. The activator doesn't like the T form, it likes the R form. And so when there's activator around, we see that the activator can bind, right? It binds here, and it has the same effect. It removes the R form from equilibrium with the T form. So now it again has the same effect. It's going to shift T into more R. So now substrate and activator are doing the same job of basically creating more of the relaxed form so that more binding can happen. So the binding of substrate and activator increases the binding. And it actually almost does away with the sigmoidal sort of aspect of it. We see that it sort of shifts it to be more of the hyperbolic type myoglobin curve that we saw before. Now there's inhibitors as well. Inhibitors actually have a higher affinity for the T form. What an inhibitor does is it binds to the T form and pulls the T form out of equilibrium with R. So that now the T form starts to, let's just call it Ti, T inhibitor. So now there's less T, and so more R shifts over to the T form. And so less binding is going to happen. And so this is how the inhibitor and the activator, through the concerted model, are actually able to do this. Now, the sequential model is a little different. The sequential model actually uh, uh, explains a little bit of also what we see with cooperative binding. The R form is more favored by the allosteric activators. The allosteric inhibition occurs by the induced fit mechanisms. There's negative cooperativity, where we see the binding of one ligand to an enzyme actually causes the next ligand to have a lower affinity. We see this with different kinds of tRNA synthetases. These things normally will put amino acids onto tRNAs. If it gets a tRNA that has an amino acid and it binds it, there's actually on this enzyme, there's 2 spots for tRNAs, but the binding of one tRNA stops the second binding spot from wanting to bind the tRNA. So that's something like negative cooperativity. Okay, I think that's probably all. We talked a little bit about sequential and in the previous lectures and concerted this time. I think that's enough for you guys to know at least the difference. There's also control via, so other than regulators, allosteric regulators, there's phosphorylation. So this is a covalent modification, and there's a few. There's serine residues, threonine residues, and tyrosines that contain these hydroxyl groups. And these hydroxyl groups are really good at forming phosphate ester bonds. And so we can make these phosphate esters. So phosphorylation by ATP will actually convert an inactive precursor into an active enzyme. And so this is an example. We've got some protein here. We activated it with ATP, and now we've got an activated protein that may be, or an enzyme that now does a job that it didn't do before, or it does a job slightly faster because it's got a conformational change that's now more conducive. So turning on or turning off of enzymes with phosphorylation can go both ways. Like we can phosphorylate it to turn it on, we can phosphorylate it to turn it off, and then dephosphorylate it to turn it back on. Like different enzymes are regulated differently. Here's an example of one of these mechanisms. This is the sodium-potassium pump. It's involved in cycling the membrane protein between the form that binds sodium and the form that binds potassium. So same protein, when it gets phosphorylated, so enzyme phosphorylated, right? It's got a phosphate group. Now it's able to pump. Now it's able to accept sodium from inside the cell and pump it outside the cell. And then now that job's done, it gets dephosphorylated, and now it's in a form where it can move potassium from outside the cell to inside the cell. And then it gets rephosphorylated again, and now it goes to sodium. So this thing goes back and forth, pumping, 2 different ions across their concentration gradients. Another example, glycogen phosphorylase. This one's actually subject to allosteric control and covalent modification. So you can see here, the phosphorylated form is actually the more active state out of the 2 T states, right? There's phosphorylase B and phosphorylase A. When they get phosphorylated, and kinases, this is just a sidebar, kinases are those enzymes that usually transfer phosphate groups. We usually call them kinases. When this thing gets phosphorylated, it is in a more active state. Now, that's one type of control, covalent control. Both of these, phosphorylase B and A, are both also subject to allosteric regulation. So you can see here, glucose, 6-phosphate, ATP, glucose, and caffeine will all cause the R state to go more into the T state. So those look like they're inhibitors, whereas AMP is an activator. That'll actually cause these things to switch into a more active state. Glucose and caffeine on the other side of this phosphorylase A, again, can be, looks like they're inhibitors for the R state. They cause T state. So, multiple types of control, and this is not all types of control, this is just giving you a tip of the iceberg type situation, but enzymes can be more heavily regulated by various types of combined covalent modifications, allosteric regulations. Sometimes they can even be created as inactive proteins called zymogens. What I'm showing you here is chymotrypsin. Now, chymotrypsin needs to be activated by specific hydrolysis of certain peptide bonds. So this gets synthesized and stored in the pancreas. Okay, it gets synthesized as a single polypeptide chain of like 245 amino acids, cross-linked by 5 disulfide bonds. This is the tertiary structure. This thing would be all folded up, and different residues of cysteines are actually connecting to each other. When secreted into the small intestine, the digestive enzyme trypsin actually goes and cleaves the polypeptide bond from the N-terminal to give active, what is it called? Pi chymotrypsin. Oh, sorry, it's right there on the right there. You can see it. So trypsin comes in, cleaves at the end of arginine here, and trypsin, by the way, goes and attacks basic amino acids. So amino acids that have those amino groups on their R chains. So it clips it there. Now it's an active enzyme, pi chymotrypsin. Now self-digestion will happen, and chymotrypsin will excise 2,4 amino acids from its own structure, its own primary sequence, to be in active form. And the reason why the body does this is because you don't want chymotrypsin, which is a protein digester. It's a protease. It cuts proteins. You don't want it doing the job where it was made. You want it doing the job in the small intestine where digestion happens. All right. So changes in that primary structure affect the changes cause changes in the tertiary structure. It was inactive, and now it's active because of those changes. All right. So let's talk about active sites. So when we talk about active sites or enzymes, we want to know how the enzyme works. So we want to ask a few questions. Which amino acids in the enzyme or on the enzyme are in the active site? Which ones catalyze the reaction? What is the relationship in 3 dimensions, in spatial relationship between these essential amino acids in the active site? What mechanisms are these essential amino acids using to catalyze the reaction? And so there's a few different, ways that we find this stuff out. And so I'm going to show you first how we kind of get the mechanism. Second, how we identify what the amino acid residues are that are important, and then we're going to talk about what the actual the catalytic or reaction is inside of chymotrypsin. So we're interested in chymotrypsin, and we're going to explore the active site. And so chymotrypsin digests proteins in the small intestine, hydrolysis of the peptide bonds that are adjacent to amino acids that have aromatic residues. So chymotrypsin goes and cleaves right after, going from N to C, right, following the amino acid chain N to C, goes right after the amino acid that's aromatic. In the lab, we also saw in the last chapter that chymotrypsin will hydrolyze esters, and we can study those in the lab, usually because they create products that are, a different color, and that makes it easy. What I'm showing you here is that chymotrypsin will actually hydrolyze p-nitrophenyl acetate into p-nitrophenolate and acetate. Okay, but when we study the formation of the product, we see that there's actually 2 things going on. We see first off that acetate doesn't get formed for a while. So the product that we eventually see isn't forming at the same time as the first product we see. We see a buildup of the p-nitrophenolate, or the para-nitrophenolate. We see that first during this, what we call lag time, and then we see amounts of these 2 actually go up together. But there's something going on in the beginning, and this tells us that there are basically 2 steps. That there is a first step where the p-nitrophenolate is actually getting removed or released, and that there must be an enzyme intermediate, an enzyme acyl intermediate, where that other portion of the protein, or in this case the other portion of the molecule, the acetate, is still attached to the enzyme. So at stage 2 then, the acyl enzyme intermediate is hydrolyzed, releasing the acetate and regenerating the free enzyme. So yeah, we see that there's a first step that's slower, and then a second step that's much faster. Now, both of these steps are what we call well, I guess we could come back and talk about it. I was going to say these are called SN1 and SN2 reactions. I'm not sure how much you guys remember from SN1, SN2 from your Gen Chem. I'm sorry, from your organic chemistry. But nucleophilic reactions, which are those that we're going to be looking at, if they take place what we call unimolecularly, then they are, or they solely depend on the concentration of one species. In this case, it could be the species of p-nitrophenyl. The nitrophenyl acetate. Then we would call it an SN1 reaction. If it actually has two molecules that are involved, then it's called an SN2 reaction or a bimolecular reaction. The significance of that for us is that SN1 reactions tend to happen pretty quickly, but the problem is we lose stereospecificity. SN2 reactions tend to be slower. They also go by different kinetics. SN1s follow first order kinetics, SN2s follow second order kinetics. But in SN2s, we see that because the molecules have to come in from a certain side, they always form the same kind of stereoisomer, and that's what we see in nature and often in these enzyme catalyzed reactions, very specific for specific stereoisomers. And so again, both of what we're going to see is the SN2 reactions, but let's come back a little bit and talk about how we know what amino acids are important. So we're going to be talking about chymotrypsin. Chymotrypsin is a serine protease. Now, a serine protease is a class of proteolytic enzyme by which a serine hydroxyl plays an essential role. And there's a couple of different, serine proteases. I can give you an example. Trypsin, chymotrypsin, elastase, all of these are serine proteases. These enzymes are found in prokaryotic and eukaryotic cells. They all use a common set of the 3 amino acids in the active site called a catalytic triad. And these consist of aspartic acid, histidine, and serine, and this is not any different in chymotrypsin. You can see that here. The serine, the histidine, and the aspartic acid. Now, the serine is going to be activated in the reaction mechanism to form a nucleophile. And so this is where they get their names, these serine proteases, because the serine is going to be the one that initiates the reaction. Now, in terms of discovering which are the active residues, we play around with it by throwing molecules at it and seeing what happens. So DIPF, which is diisopropyl phosphofluoridate, basically inactivates chymotrypsin. We know that it inactivates chymotrypsin because when you put it in there, chymotrypsin stops working. It reacted with the serine number 195, and so it verified that that serine was in the active site, if you mess with that serine, you turn off the enzyme. So they figured out the serine that way. Histidine 57 they figured out was critical also. It can be chemically labeled by something called N-tosylamide L-phenylethyl chloromethyl ketone. Yeah. Anyways, when they do that, basically, again, they can knock out the activity and figure out where the that histidine was important, and that you can't, that anything that will react with that histidine will kind of mess up the action. Those two have to be near each other, the serine and the histidine. Aspartate's involved also. It just needs to be near the catalysis site. You can see here that the aspartate actually has a role in sort of hydrogen bonding with the histidine, sort of holding it in place. X-ray crystallography has shown the definite spatial relationship amongst these guys. We can also see the protein backbone here with some amino acids that were talking about, kind of shown right here in the active site. Okay, let's talk about how this reaction happens. So let's jump here. So there are two other kind of terms that we use when we're talking about reactions. We talk about nucleophilic substitution reactions. Now, nucleophiles are molecules that that are electron rich and are seeking sites of positive charge. And so they're seeking nucleuses. And so what we're going to see here is that this oxygen right here is going to be a nucleophile. It's going to be an electron-rich area seeking out a positive area. And the positive area is going to be this carbonyl carbon down here. This polar bond, this double bond here is very polar, and the carbonyl has got that positive charge there. Now, what's going to also happen is that this histidine is going to pull this hydrogen off of this oxygen in what we're going to call general base catalysis. Now, before I go on with that, let's talk about the other type of phile. So in contrast to nucleophiles, there's electrophiles, and electrophiles are electron-seeking substances. They tend to be electron poor, and they tend to be trying to interact with electron-rich places or negatively charged places. But this is going to be a nucleophilic substitution reaction, so we're going to get the serine being a nucleophile. Now, it's also worth mentioning here that histidine is going to play a role in both the general base catalysis of this. Now, let me remind you what the other version of catalysis is. There's specific acid-base catalysis, and this means that specifically there's hydrogen ions or OH hydroxide molecules, hydroniums or hydroxides that are being added. And so catalysis with these guys means you add H2SO4 to make it go faster, or you add sodium hydroxide to make it go faster. Then there's general base catalysis. General base catalysis means that we're going to have hydrogen donations or acceptors. Donors, acceptors. So these are not going to be actual hydroniums or hydroxides. And so histidine plays the role here of a base catalyst. It's going to accept a hydrogen from this oxygen. Okay. Actually, we didn't mention anything about the specificity, but it's neat to mention here that the specificity of this enzyme's active site is such that these amino acids are arranged in just a way so that it knows when there's an aromatic residue there and when there isn't one. And that's kind of how these enzymes are so good at being able to only cut bonds that are adjacent to the one, to aromatic residues. This is why this first this reaction can be I don't know what I was trying to say. I'm sorry. I think I was thinking of some neat way of saying how enzymes are cooler than they are, but then I started getting lost in thinking about how the enzyme really doesn't think about anything. Sorry about that. Okay, so specificity is determined by the active site's shape, of course, and interactions with the R groups, and of course some of these residues are here in the active site just to interact with the part of the molecule that they're meant to cut as a part of seeing if it's the right molecule. Okay, so let's say the process starts with the binding of our molecule into the substrate binding pocket here of chymotrypsin. It has a hydrophobic hole where the substrate kind of fits in. The preferred substrates for this molecule are going to be bulky hydrophobic groups like phenylalanine, right? Aromatics. If it's got an ionized side chain like glutamic acid, for example, it'll exit quickly. Just like water would from anywhere that was oily, it's just going to bounce right out. When the proper molecule actually binds, and so in this case, this is the molecule down here, we're going to see the change of the enzyme's shape change just a little bit. Subtle change shape that's actually going to start the catalysis. Now, since catalysis only starts when the proper substrate binds, this is part of the reason too why we see such good specificity. Okay, so once it binds, the shape kind of changes. The slight change actually involves the triad here, the catalytic triad. The aspartic acid, histidine, serine, they all sort of shift into position, and then everything starts. So the shift of the negatively charged aspartic acid towards the electron-rich histidine ring sort of favors the abstraction of a proton by the histidine from the hydroxyl. So all of that happens together. The aspartate's not being shown in this picture, but I did show it here. So this is step one right here. So because the aspartate sort of pulls on this guy's hydrogen, this nitrogen decides, hey, I'm going to go and take this hydrogen off the oxygen. And that gives this oxygen the electron-rich environment that it's going to use to go be a nucleophile. So then it goes and makes a nucleophilic attack on the molecule, whatever the molecule is, resulting in a, so first we get this very reactive alkoxide ion, and then that goes and reacts with our polypeptide chain. So then the nucleophilic attack happens on the peptide bond of the phenylalanine, and the reaction breaks the peptide bond, and then two things happen. So we can see here the bond. We see here, hold on, I'm looking right here. We form a bond, so we've formed this bond here, and then the bond right here, the peptide bond, breaks, and that nitrogen then grabs this hydrogen, and so and then it runs off. There's actually two things that happen there then. So here, so referring to the histidine real quickly, histidine here was a base, acted as a base catalyst. It pulled this hydrogen away so that the serine could do its job. And then here, after the serine had done its job, it donated this hydrogen. It donated it to the leaving group, so it played an acid catalyst role. So this is the general acid-base catalyst. Now, what's interesting about histidine playing an acidic role here is that histidine wouldn't have been able to play this role under normal physiological conditions, because the pKa for histidine is around 6. So that means the histidine side chain at around 6 would have lost its hydrogen. But because of the situation here in the active site, the neighboring, the closeness of these amino acid residues to each other, histidine at a much higher physiological pH is still able to play an acidic role. It's pretty neat. Okay, so first, the original polypeptide is free. It exits the active site. Second, the end containing the phenylalanine is actually covalently linked now to the oxygen. So this is the rest of the amino acid chain that had the phenylalanine still attached to it. Now, it's bound to that oxygen. So that was now we're in the second phase where we have to regenerate the enzyme. Okay, so the second phase of the catalysis is slower. It requires that the covalent bond between the phenylalanine and the serine's oxygen be broken. It starts with the entry of a water, so we get a water molecule coming in, and again, nitrogen is going to play, or at least the histidine is going to play a base catalysis role. So the water enters. It's attacked in a fashion similar to that of what the serine did the first time, but this time by the phenylalanine. And so the phenylalanine is going to sort of pull its hydrogen off, creating a more nucleophilic oxygen. That oxygen is going to nucleophilically attack the carbonyl carbon again. And once that happens, it attacks the phenylalanine serine bond. It releases it and replaces the protein on the serine. So you can see that that hydrogen that the phenylalanine gained is just going to basically be given back to the serine, and then all of our stuff down here is going to float off into space. So the second peptide gets released. All right, so histidine activated the serine to be a nucleophile, and then histidine activated the water to be a nucleophile. Now, there's a long list of serine proteases. They're grouped into two broad categories: those that are chymotrypsin-like, and those that are subtilicin-like. Can never say the word properly. Subtilicin-like. Same mechanism of action, including the triad. They are just not related to each other in any other sequence. They appear to have evolved sort of independently of each other, kind of convergent evolution. So that's kind of neat. The serine proteases, enzymes cut adjacent to specific amino acids, and yeah, chymotrypsin, trypsin, I can't say it again. Elastase. I said a few of them earlier. Now there's a few other types of proteases. There's cysteine proteases. These are sometimes known as thiol proteases because they use the thiol, the SH group found on the cysteine residues. Very similar mode of action to the serine proteases. There's also caspases. These are a family of cysteine proteases that play important roles in the body. They function in like apoptosis and necrosis and inflammation and other things. There's metalloproteases. These are enzymes whose mechanisms for breaking peptide bonds involves a metal. So I'm going to show you an example of how some of the metals work in there. Let's see, threonine proteases. There are protease inhibitors. There's a lot of molecules actually that block the catalytic activity of proteases. Variety of forms, biological and medical uses, as you can imagine. They can be suicide inhibitors. They can be transition state inhibitors. They can denature them. Anyways, there's lots of ways of inhibiting proteases, just like other enzymes. All right. Okay, let's see what we have. Speaking of metal ion catalysis, so metals are often called Lewis acids, or metal ion catalysis is called Lewis acid base catalysis. But metals can often be Lewis acids in that they are electron pair acceptors, and this is usually because they have open D shells that are available for putting electrons into. So like manganese, zinc 2+, or manganese 2+, magnesium 2+, zinc 2+. These are often essential for many enzymes. Carboxypeptidase A, for example, that I'm showing you here, requires a zinc 2+. It's actually complex to the imidazole side chain of the histidines. Histidine 69, I think it is, and 196, and then the carboxylate side chain of glutamate 72. And what it does is it activates the carbonyl group for nucleophilic acyl substitution. So by being positive, it kind of pulls on the negatives, making this even more positive for a nucleophilic substitution. So enzymes are pretty specific. We talked about that a little bit, and just in their active sites and what they recognize and what they bind with. They can be absolutely specific, meaning that they only catalyze a unique substrate to one product. They can be relatively specific in that they can catalyze structurally related substrates to give related products. They can be stereospecific in that they only react with one isomer. Here's an example of that right here. The hydration of, as an example, hydration of like a cis alkene to give an R alcohol but not an S alcohol, for example. They can make very specific very specific molecules. They can also bind specific isomers, and here is an example of binding a specific isomer. The binding sites would be, in this case, asymmetric. So if the other molecule, right, tried to fit in here that had the little triangle at the bottom and the little kind of that on this side, that wouldn't match, right? The D version and the L version would not be superimposable. So they're specific in that way. Um, the transition state actually has an interesting role to play here. So enzymes provide an alternative pathway, right? Lower activation energy. Um, the transition state is actually something that's an intermediate between the substrate and the product, and the transition state usually has a different shape from either of the two, substrate or product, and the transition state is actually used in something called what did they make? Enzymes. So transition state analogs are something that people have made for a long time. They're compounds that mimic the transition state of an enzyme reaction. And it was in like 1969 that somebody proposed that if you took a transition state to an enzyme, or of a substrate in an enzyme, and you introduced it into someone's body as an immunogen. So an immunogen is something that triggers an immune response, and usually it means that your body will make antibodies against it. So the idea was, if you take a transition state for a reaction, like in this one right here. So if we take L-proline that can be turned into D-proline by an enzyme, and we copy this transition state and we make an analog to that, and then we introduce it into your body will actually produce antibodies that are catalytic. They theorized this, and it actually was true. So the antibodies, they're called enzymes. The antibodies that are produced against a transition state analog. So this guy right here is like a transition state analog to whatever enzyme does this job. Yeah, this guy looks kind of like that, but they're different. If you put this into your body, your antibodies that get made against this, what it thinks of as an immunogen, would create antibodies that have catalytic activity similar to the naturally occurring enzyme, and that antibody would actually turn this molecule, if it saw it, into this molecule. It would have that catalytic activity. I don't know what the point of that was, of what they did. I think it was somebody's neat understanding of what they were looking at, that the enzyme's active site that actually does the catalysis could be mimicked if they created basically by antibodies. Antibodies made against transition state analogs are catalytic for those, and end up, you can use them almost in place of the enzyme. I don't know if they lose all their specificity, though. That might be the rest of the protein, is there to make sure you're only doing that job to the right molecule, and when you just take kind of like a free-floating active site, you may lose a lot of that specificity. Okay, coenzymes. Let's talk a little about coenzymes. So coenzymes are non-protein substances that take part in enzymatic reactions. They're regenerated at the end of a reaction. Metal ions can behave as coordination compounds. We saw that a little bit with the zinc and the iron. Zinc in that enzyme I just showed you, and iron, of course, in hemoglobin. Usually, organic coenzymes are like vitamins. Of course, there could be metals as well. I mentioned that. Usually, they call coenzymes that are metals cofactors. I don't know. The terminology changes. It seems like your book is calling everything a coenzyme, and it's just differentiating organic coenzymes from the non-organics. Non-organic coenzymes in the old books I used to teach out of were called cofactors. So coenzymes can be vitamins and their derivatives. These organic coenzymes are often involved in oxidation reduction reactions, like the one I'm showing you here. This is NAD. Well, actually, I'm not sure which form it is that we're looking at here. NAD will get, let's see, so we're looking right down here. Yeah, so this is NAD+, and it can become NADH when it gets reduced, and so you can see that this is the part of the molecule that's actually getting reduced. You see additional hydrogens there, and there's an additional hydrogen, or it gained some electrons. So it gained an H and some electrons. So NAD into NAD+. We're going to go over NAD a lot when we talk about catabolism of sugars. Let's see. Structure-wise, it consists of a nicotinamide ring, an adenine ring and some sugar phosphate groups linked together. I don't know if there was anything else I wanted to say about that. There are some other examples. I got another example here. B vitamins. Pyridoxal, pyridoxamine, pyridoxine, and their phosphorylated forms, which would be these guys. These guys are also the B6 vitamins. They're coenzymes involved in different amino transfers or amino group transfers. You can actually see that one here. There's a transaminase here. And two stages. First, looks like we have pyridoxal phosphate attached to pyruvate, and then yeah, there's some stuff going on there. Pyridoxal and pyridoxalamine phosphates, again, just involved in the reactions of some of these amino transfers. Not a reaction I want you guys to memorize or anything, just some examples of some coenzymes. Let's see. What else can we talk about for enzymes? I think that's kind of it for the chapter-wise. That's all the material. I gave you guys more info than the book had in regards to like the other types of proteases. But the serine protease is the one that we focused on, and the chymotrypsin mechanism of action is the one that we looked at. Let's see. If the coenzyme is very tightly or covalently bound, it's actually referred to as a prosthetic group. So the heme in the hemoglobin is a prosthetic group. If it was an enzyme, it could have been called a proenzyme. I'm sorry, if hemoglobin was an enzyme, it could have been called a coenzyme, the heme. Enzymes without their cofactors are inactive and referred to as apoenzymes. Apoenzyme. So inactive enzyme that's missing its cofactor. Enzymes containing all their cofactors are called holoenzymes. Holoenzymes. Biotin, coenzyme A, flavin, so FAD, NAD, pyridoxal phosphate, tetrahydrofolate, thiamine pyrophosphate, potassium ion, magnesium, manganese, molybdenum, nickel, selenium, zinc. A lot of these are cofactors. Okay, that should do it. So that is all the material for this week. That basically wraps up our first sort of module, or let's see, looking on Canvas here while I talk to you guys. So that takes care of our if you go to the modules, Module 1 and Module 2 and Module 3. So Exam 1 is ready for you guys. Yeah. So good luck on that. Definitely, put as much detail as you think you need to. If you've got a short answer that says it all, then don't feel that you need to write more. I'm not looking for wordiness. I'm looking for accuracy. All right. So then, starting next week, we'll look at carbohydrates and lipids. We're going to look at membranes. We're going to look at nucleic acids and DNA replication, and then we'll save for the week after that protein synthesis, sugar catabolism, and photosynthesis. So that'll be fun. All right, guys. Don't forget about the quiz that I have up for this chapter as well. And see you guys next week. Stay healthy.